I wonder why some people say biology is very, very difficult. This video is going to help you to understand concept and cells or cell specialization. So the question is saying figure 11.1 shows two liver cells as seen under a light microscope. So let me not forget, if you're new to this channel, subscribe and share this video to your friends. So we have structure A. This structure A, it is called the cell membrane. Then as for structure B, this one is called the nucleus. So in an exam, if they ask you to give them the function of the cell membrane, you are going to say a cell membrane selects what goes in and out of the cell. Then as for B, which is the nucleus, we are going to say this nucleus stores genetic information in form of the chromosomes, meaning the chromosomes are found in the nucleus and they are responsible for storing genetic information. Then apart from that one, you can also say the nucleus controls all the activities which happens within the cell. Question A. Name the structures labeled A and B. So as for A, we are going to say the cell membrane. Then as for B, we are going to say the nucleus. Now, not only those structures are found in an animal cell or the liver cell. We also have these small structures which are called the cytoplasms. So we can say cytoplasm. Now the function of the cytoplasm is just the site of where all the chemical reaction occurs. Or we can just say the site for chemical reactions. Then as for B, which is question B, state two functions of liver cells. Now since these two cells, they were taken from the liver, meaning they are going to perform the same function of the liver. So the first one, we are going to say cells. Now since these two cells, they were taken from the liver, meaning they are going to perform the same function of the liver. So the first one, we are going to say when there is a high level of blood sugar in your body, you're going to find that the pancreas is going to secrete what we call the insulin. The insulin is just the hormone. And the work of this insulin is to convert excess glucose or blood sugar into glycogen. This glycogen is just the form in which carbohydrates are stored in humans. Now, to answer this question, we are going to say the liver cells, they convert excess blood sugar into glycogen. That's the first answer. Then the next one, when in your body you are having excess amino acids, those amino acids, they are going to undergo what we call deamination. So in this process of deamination occurs, you are going to find that urea is going to be produced. So we can see that urea is made in the liver, not in the kidney. So to answer this one, we are going to say deamination of excess amino acids to produce urea. Then apart from that one, you are going to find that when stress glands produces the hormones and those hormones are being used up by the body or by the target organs, you are going to find that those used up hormones are going to be destroyed by the liver. So in short, we are going to say the liver cells destroys used up hormones. Then apart from that one, I can also add the last one where we can say bile production. So the bile is produced from the liver. Let's see the next one, which is C. Give three ways in which a plant palisade cell differs from a liver cell. So as you can see here, the liver cell is having the structure or the general structure of an animal cell. Meaning here they are just telling us to differentiate between an animal cell and a plant cell. So the first one I'm going to say, the palisade cells, they have the chloroplast or chlorophyll, while the liver cells don't have. Then number two, we are going to say the liver cells, they have the temporal vacuum, whereas the plant palisade cells, they have the permanent vacuum. Then the last one we are going to say, the palisade cells, they have the cell wall, whereas the river cells, they don't have. So those are the questions. Apart from that one, you can also include that, like in terms of the shape. So the palisade cell, it has the fixed shape, whereas the river cells, they have an irregular shape. Then let's come to D. This one is very, very important for us for D. In figure 11.1, the actual height of the cells along the line XY is 0.03 millimeters. Calculate the magnification of the drawing, assuming that the size of the actual cell is 0.006 millimeters. Now the first thing here, we need to know the formula for calculating the magnification. So I'm going to say magnification is going to be equal to the size of the drawing. So we are going to say the size of the drawing. Then we can say divided by the actual size of the cell. So the actual size of the cell. Now, to answer this one, we have been given the height of these two cells from X up to Y, which is approximately 0.03 millimeters. Meaning this one, it is the size of the drawing. So we are going to say the size of the drawing here is just going to be 0.03 millimeters. Then when it comes to the actual size of the cell, it is this one here, which is 0.006 millimeters. So what we are going to do here, millimeter and millimeter, they are going to cancel each other. Now mind you, in biology, you are not required to use a calculator. So what we are having here is a division. So what we are going to do here, we are going to say, this number here, which is 0.03, I'm going to convert it into a fraction. Then I'm going to obtain something like 3 divided by 100. Then divide. This one is 0.006. If I convert this one into a fraction, I'm going to have 6 divided by 1,000. Then from there now, the reciprocal of division is the multiplication. So I'm going to obtain 3 divided by 100 multiplied by 1,000 divided by 6. So 2 zeros are going to be cancelled with these 2 zeros. Then 3 multiplied by 10, I'm going to obtain 30. So 30 divide. 1 multiplied by 6, so I'm multiplying 1 multiplied by 6, I'm going to obtain 6. Then 6 into 30, I'm going to obtain 5. Now, since here we are calculating the magnification, by definition, magnification is the number of times the cell has been magnified. So usually magnification doesn't have the units, but we include x to represent the number of times the cell has been magnified. So in including x, you can include the x in front of the number or at the end of the number. So what we are going to do here, we are just going to say magnification is equal to x5. Next question. It is saying figure 5.1 is a photograph of the center of a plant root. So we have structure J. So this structure J, it is called the phloem. So phloem vessel. Then structure K. This one is called the xylem vessel. Then when it comes to L, this one L is just the collection of J, which is the phloem, and K, which is the xylem. So we are going to refer L to the vascular bundles, because vascular bundles consist of the phloem vessel and also the xylem vessel. Question A, Roman number 1. Identify each of the tissues labeled J and K. So we are going to say, as for J, it is going to be the phloem tissue. Then as for K, it is going to be the xylem tissue. The next one, Roman number 2. State the term used to describe the group of tissues labeled L. So the group of tissues labeled L, we are going to refer to these as the vascular tissues or the vascular bundles. Then as for Roman number 3, state 2 ways in which the structure of a cell in tissue K will differ from a cell in tissue J. So tissue K, it is the xylem. Then tissue J, it is the phloem. So the first thing here, I'm going to talk about the functions. So as for K, which is the xylem, it transports water and mineral salts. Then as for J, it transports manufactured foods or sucrose. So to differentiate these two, I'm just going to say K transport water and mineral salts, while J transport sucrose. So in this part here, if you don't want to include sucrose, you can say manufactured foods, or you can say sucrose stroke amino acids and potassium ion. So the next difference, we are going to say J, which is the phloem. This one is made up of living cells. So we are going to say J is made up of living cells. Then when it comes to K, which is the xylem, it is made up of dead cells. So we are going to say, well, K is made up of dead cells. So let's move on to the next one. So the next one is saying, figure 8.1 shows a photograph of some red blood cells. So the first question, which is question A, Roman number 1, is saying, state the function of the red blood cell. State the function of the red blood cell. So the function of the red blood cell, it is to transport oxygen and antigens around the body. So the antigens, they are found on the cell membrane of the red blood cells. Then the oxygen, it is going to be trapped by the hemoglobin. So the hemoglobin is found on this part, which we are going to call it as the biconcave in shape. So that's the answer. Roman number 2. State 2 ways in which a red blood cell is adapted to its function. So the first one, I'm going to talk over the biconcave in shape. So this part here, this part, we call it as the biconcave in shape. So we're going to say the presence of biconcave in shape to create more room for the carrying of hemoglobin. So next adaptation, we are going to say the presence of hemoglobin. Now this hemoglobin attracts oxygen. So the presence of hemoglobin for the attraction of oxygen. So those are the answers. The next one, state the name of a component of blood that is responsible for clotting. So the component of blood which is responsible for the clotting, it is called the platelet. So you can say platelet or platelets. Now platelets can be able to clot the blood in the presence of vitamin K. So when the body is lacking vitamin K, you're going to find that you're going to have difficulties in blood clotting. Apart from that one, if you're lacking the clotting factors or you are being affected by hemophilia. So those are the conditions. Then as for question C, Roman number 1, it is saying, state the name of the disease caused by the blockage in the arteries supplying the heart muscle. So this disease, it is called the coronary heart disease. Saying it is called the coronary heart disease. So that's the answer. Then as for Roman number 2, state 3 risk factors that increase the risk of a blockage in the artery supplying the heart muscle. So the first one, we are going to say excess intake of alcohol. So when you are taking too much alcohols, the coronary arteries are going to be blocked. Therefore, you are going to suffer from the disease called coronary heart disease. So the first one, we are saying excess intake of alcohol. Then apart from that one, we are going to say smoking. So smoking also causes the blockage of the coronary arteries. Then apart from that one, we are going to say excess intake of fats. So if you are eating food containing excess fats, you're going to find that the coronary arteries, they are going to be blocked, thereby preventing blood supply to the heart. As the result, you're going to be affected by the disease called the coronary heart disease. So that was the last one. Thanks for watching and see you next time in the next video.